Neuropathology of Alzheimer's disease, memory symptoms,
by Andrea Milbury
and Cheyenne Wilson.
What is Alzheimer's disease? Alzheimer's disease is the most common form of dementia and is a neurodegenerative disease that slowly increases in severity over time until a person's brain can no longer withstand it and it dies. It was first described in 1907 by Alois Alzheimer and is characterized mainly by memory impairment and cognitive deficits. Baird in 2015 states the most clinical cases of Alzheimer's disease last 8 to 10 years. However, the duration of the disease may be as short as 1 year or as long as 25 years. The cause of Alzheimer's disease can be broken down into 3 categories: neuropathological, which are the hallmark causes of the disease, such as amyloid beta plaques, aka senile plaques, and tau neurofibrillary tangles. Familial, which includes the effect of ApoE4 gene and makes up approximately 1/4 of all Alzheimer's disease cases. And environmental, which is still currently being researched but may include formaldehyde concentrations in the body and insulin deficits. Sleep deprivation is also an environmental factor, which we'll be going into further detail about later on. Amyloid beta plaques. Typically, in a healthy brain, proteins are slowly processed on the extracellular membrane of a neuron. In this process, 2 enzymes, alpha secretase and gamma secretase, cut amyloid precursor protein from the membrane. These fragments are now floating in the extracellular fluid and are said to aid neuron activity. However, the brains subjected to AD have different enzyme than alpha secretase that is making the first cut. Instead, it is beta secretase. Its cut, combined with the gamma secretase cut, makes very small fragments of the amyloid precursor protein, which are called beta amyloids. The beta amyloids aggregate together to form insoluble amyloid plaques, which are toxic and enhance neurodegeneration. The small fragments can also get into synaptic clefts of the neurons and prevent synaptic transmission, thus decreasing brain networking and eliminating pathways to memories and destroying the memories themselves. So here we have an image capturing this process. As you can see, the amyloid precursor protein has made its way to the cell membrane. The enzyme beta secretase then makes its first SNP. Do note that the SNP is irregular in comparison to the regular alpha secretase functioning. Next, gamma secretase makes the second SNP, and the amyloid precursor protein, along with the beta aggregate, are sent floating into the extracellular space. These floating aggregates can cause the infamous amyloid beta plaques, or, as mentioned, can clump in the synaptic clefts of neurons. Many researchers support the idea that the deposition of amyloid beta plaques is not the major contributor to the symptoms of Alzheimer's disease, despite the huge impact it has on neurodegeneration. However, laboratory and animal research proposes the idea that amyloid beta impairs long-term potentiation, important for the consolidation of memory. In addition, experimental data suggests that amyloid beta toxicity groups impairs spatial memory, but only when combined with the occlusion of bilateral common carotid arteries. Also, unlike what is seen in tau pathology, the amount and location of amyloid beta plaques does not actually matter, as supported by several autopsies.
Tau neurofibrillary tangles. The tau protein is a protein that is highly associated with microtubules, a component of the cell that is essential for exoplasmic flow or transport that is necessary for neuronal activity. In normal humans, tau proteins, with the help of 2 other proteins, help stabilize microtubules, allowing nutrients and other components to be transported throughout the neuron. However, in Alzheimer's disease, tau proteins are abnormally hyperphosphorylated, causing them to separate from the microtubules and join together to form neurofibrillary tangles. In effect, microtubules fall apart, inhibiting exoplasmic transport, thus destroying the cell and causing neurodegeneration. Tau neurofibrillary tangles and memory symptoms. Tau neurofibrillary tangles are strongly correlated with multiple memory symptoms, predominantly spatial memory impairment. Research supports that the medial temporal lobe, as well as a distributed set of cortical regions, is vital for memory functions such as encoding, consolidation, and retrieval. However, in Alzheimer's disease, cell death, primarily in the medial temporal lobe, arises as a result of tau neurofibrillary tangles, disrupting memory function and retrieval. This is supported by a study done by Fu et al. In 2017. The researchers looked at the memories of mice that were injected with tau in comparison to a control group to look at the effects of mature tau on learning and memory. Both groups were taught a maze and were made to redo the maze after 4 days, again after 14 months, and one last time after 30 months. It was found that in tau-injected mice, there was significant regression between 14 and 30 months, especially in comparison to the control group, and they exhibited both spatial learning and spatial memory deficits. Because these deficits were found only in the tau-injected mice, and mainly after 30 months, the researchers concluded that mature tau pathology was the main contributor. Tau neurofibrillary tangles has also been shown to impair episodic and semantic memory. Gray matter volume is said to be negatively correlated to tau neurofibrillary tangles. Therefore, tau pathology reduces gray matter volume, and these reductions can lead to semantic or memory impairment depending on the location. For example, researchers have established that the entorhinal cortex and the hippocampus play a role in episodic memory, and in 2013 it was discovered by Herney et al. That reduced gray matter volume in these areas on both sides of the brain results in episodic memory impairment. Similarly, reduced gray matter volume in the medial parahennial cortex, which is associated with semantic memory, is related to poor semantic memory performance, but only when the reduction is on the left side and in conjunction with gray matter volume reduction in the entorhinal cortex and hippocampus.
APO is a gene that encodes the apolipoprotein E molecule, which was originally known for its metabolic processes of breaking down, transporting, and binding fat molecules in the brain and liver. Now it is known as a pleiotropic molecule, meaning it affects multiple ways by the gene, which many important functions including lipid transport, amyloid beta trafficking and disposition, synaptic function, intracellular signaling, and more. In humans, there are 3 common allele variations of the APO gene, and they are E2, E3, and E4. Each have their own effect on the disposition of amyloid beta. APOE4 had the highest influence on the amount of amyloid beta plaques. In fact, a research by Liu et al. In 2014 used immunohistochemistry and saw that 40.7% of carriers of the APOE4 allele had an abundance of senile plaques in comparison to 8.7% who had them, but did not carry the allele. The likelihood of developing tau neurofibrillary tangles is also studied to be more likely when possessing the APOE4 allele. In fact, the toxicity of amyloid beta plaques depends on the amount of tangles. As discussed prior, the abundance of these tangles and toxicity of the plaques is harmful to neural activity and enhances memory loss. APOE4 is also lower in cerebrospinal fluid, which we will see the effects of in an upcoming slide. APOE4 allele also correlates with an increase in synaptic dysfunction. The APOE4 allele carrying mice in their lab had less dendritic spine density on postsynaptic neurons, thus reducing synaptic transmission and neuroplasticity. In addition, due to the importance of hippocampal neurogenesis in structural plasticity and maintenance of brain networks, dysfunctions caused by early onset of Alzheimer's disease may make neurons more vulnerable to the disease and more memory impairments. The gene APO is responsible for the conservation of the regions in the hippocampus. In a study, the E4 allele inhibits hippocampal neurogenesis, which results in learning and memory deficits in lab mice, thus supporting APOE4 being responsible for the impairment of neurogenesis, which increases the vulnerability to Alzheimer's disease.
The role of astrocytes. Astrocytes are a rare type of glial cell found within the central nervous system that are said to maintain structural, metabolic, and guidance support for neurons. Neurodegenerative diseases like Alzheimer's disease, as well as other harmful processes such as injury or infection, cause a phenomenon known as astrogliosis. This phenomenon occurs when there is an increase in astrocytes, known as reactive astrocytes, triggered by the destruction of nearby neurons, and may result in a number of neuropathological changes, such as excitotoxic glutamate release or blood-brain barrier breakdown. It is also correlated with both amyloid plaque and tau neurofibrillary tangles. Connection to tau pathology. Researchers believe that astrogliosis is more directly correlated with tau neurofibrillary tangles than amyloid plaques. This is because as the disease progresses, astrogliosis responses change in a linear fashion with the neurofibrillary tangles. That is, as tau neurofibrillary tangles become more abundant, dense, and distressing, astrogliosis increases as well. In addition, research shows that astrocytes have a tendency to overexpress tau, which causes problems with certain cellular functions, for example, the distribution of proteins and other molecules, and may result in detrimental effects such as the degeneration of the blood-brain barrier and abnormal protein expression, sometimes even cell death. Connection to amyloid plaques. As previously mentioned, amyloid beta plaques are caused by the involvement of beta secretase in the fragmentation process. Beta secretase APP cleaving enzyme, or BASE1, specifically is the main beta secretase involved in this process, and is especially seen in brains subjected to Alzheimer's disease. Stressed astrocytes and reactive astrocytes near amyloid plaques contain the BASE1 gene. In effect, these astrocytes produce and may even be the primary source of amyloid beta in the brain during the progression of Alzheimer's disease. Therefore, astrocytes have a major contribution to the progression of Alzheimer's disease due to their effects on tau neurofibrillary tangles and amyloid beta plaques.
The role of cerebrospinal fluid. Bodily processes all require energy consumption and efficient byproduct removal. In most parts of the body, lymphatic vessels get into the tiny spaces between cells to collect their wastes. However, there are no such vessels amongst the brain cells, even though it uses 25% of our daily energy supply and makes the waste byproduct from it. Researchers found that the removal of these wastes is due to cerebrospinal fluid, or CSF, surrounding the brain. CSF was shown to seep into the depths of the brain along the blood vessels. The waste diffused into the CSF similarly to how it would into the lymphatic vessels in other locations of the body, and then from the CSF they will travel to the cervical lymph nodes in the base of the skull. Despite the abundance of research, the driving force for this process is still elusive. Interestingly, this waste collection process only happens while humans are sleeping. This is where CSF has an involvement with Alzheimer's disease. Unfortunately, the elderly are more likely to experience interruptions in sleeping patterns and are known to have increased sleep deprivation. As mentioned above, carriers of the ApoE4 gene also have reduced amounts of cerebrospinal fluid. Thus, they have less time for the elimination of waste, specifically amyloid beta aggregates, one of the hallmark causes for Alzheimer's disease. This waste evacuation process depends on the diffusion of CSF into extracellular spaces, or ECS. Alzheimer's disease patients have diffusion impairments of the CSF to the ECS. This prevents the CSF from immersing into the brain's depths, inhibiting the efficient removal of amyloid plaques.
The role of dopaminergic cell death. The ventral tegmental area is located in the brain near the substantia nigra par compacta. This area is highly associated with dopamine and projections to the hippocampus, a part of the brain majorly associated with memories. Recently, Nobili et al. Proposed the idea that dopaminergic cell death within this area is located with memory deficits, and provide evidence with the use of lab mice. Interestingly, the researchers found that long-term potentiation was impaired at approximately 6 months of age, when there is significant ventral tegmental area cell death and a reduction in dopamine outflow by the hippocampus. This supports the idea that dopaminergic cell death within this area is accompanied by memory deficits. Despite what may have been previously thought, there are multiple contributors to the memory impairments seen with Alzheimer's disease. We saw that amyloid beta, tau, and the ApoE4 gene had deterring effects in the brain. Astrocytes, cerebrospinal fluid, and dopaminergic cell death play a role in the disease's progression as well. However, the knowledge about the neuropathology of AD and its memory symptoms is still incomplete. because researchers are still discovering more and more about this topic every day. Thank you.